reactions of heme synthesis. In total, there are 8 reactions for heme synthesis. It is both mitochondrial and cytoplasmic. The starting point and ending point both are in mitochondria. So the first step is the condensation of succinyl-CoA with glycine to form delta amino levulinic acid, which is otherwise named as ALA. Because delta amino levulinic acid is being synthesized, the enzyme is ALA synthase. Now for this ALA synthase, pyridoxal phosphate acts as a coenzyme. Now this delta amino levulinic acid enters cytoplasm. In cytoplasm, 2 molecules of ALA combine together to form one porphyrinogen. So this porphyrinogen is one pyrrole ring. So because porphyrinogen or otherwise PBG is getting synthesized, the enzyme is PBG synthase, and also in this reaction 2 water molecules are removed. So the enzyme can be ALA dehydratase, that is the removal of water molecule. Now because heme is a tetrapyrrole ring, now 4 porphyrinogen molecules, porphyrinogen is single pyrrole ring, so 4 such molecules join together to form a linear tetrapyrrole structure, that is hydroxymethylbelane. Now in this reaction 4 ammonia molecules are removed, and the enzyme, because HMB is getting synthesized, the enzyme is HMB synthase, or because these ammonia molecules are being removed, the enzyme can be PBG deaminase. Now this HMB molecule, that is hydroxymethylbelane, has 2 fates. One, in the presence of the enzyme UPG1 synthase, it spontaneously undergoes condensation to form uroporphyrinogen 1, or it undergoes, in the presence of UPG3 co-synthase, it forms UPG3, uroporphyrinogen 3. Now because this is a type 3 porphyrin, these substitutions are arranged in an asymmetrical manner. Now this uroporphyrinogen 3, in the presence of the enzyme decarboxylase, that is 4 carbon dioxide molecules are removed, because this acetyl gets converted into methyl. Acetyl is getting converted to methyl. So 4 acetyl molecules gets converted to methyl molecules, and with the removal of the 4 carbon dioxides in the presence of uroporphyrinogen decarboxylase, it forms coproporphyrinogen 3. Now this coproporphyrinogen 3 enters mitochondria. Now the coproporphyrinogen 3 enters mitochondria, and in the presence of the enzyme CpG oxidase, this coproporphyrinogen 3 gets converted to protoporphyrinogen 3. Now what happens actually here is the substitutions methyl, 2 substitutions of this group, the methyl, they get converted into vinyl. The 2 substitutions, they get converted to vinyl, and molecular oxygen is required for this reaction. So that is why the enzyme is CpG oxidase. Now the next reaction is in the presence of PPG oxidase enzyme, protoporphyrinogen oxidase. This protoporphyrinogen 3 gets converted to protoporphyrin 9. So what happens in this is the methylene bridges which are present in the protoporphyrinogen 3, they get converted to methinyl bridges. So these methinyl bridges are the bridges which combine the pyrrole structures. Finally in the heme there are 4 pyrrole rings, and these 4 pyrrole rings are joined together by these methinyl bridges. So the conversion of methylene to methinyl bridges occurs in this reaction in the presence of the enzyme PPG oxidase. Now in the last step to this porphyrin ring, an iron gets attached in the presence of the enzyme heme synthase, because heme is getting synthesized finally, or because iron is getting attached, the enzyme can be ferrocalatase. So finally the heme gets synthesized when an iron gets attached to this protoporphyrin 9 molecule in the presence of the enzyme heme synthase or ferrocalatase. Now this whole reaction of heme synthesis can be remembered with the help of the mnemonic delta P U C pro H, because of the products that are getting formed. Delta means delta amino levulinic acid, P is porphyrinogen, U is uroporphyrinogen 3, C is coproporphyrinogen 3, pro is protoporphyrogen 3 and protoporphyrin 9, and H is heme. In between of this there will be one molecule HMB, that is hydroxymethylbilane. Now this is a linear molecule which spontaneously condenses to cyclize or cyclizes to form the uroporphyrinogen molecule.